to the environment. What is also worthy of noting, NIR. Those are antibodies that have not been approved by the FDA. So the rest of these are approved antibodies. So once the antibiotic is administered to the hog, there's a pathway. We understand from several studies that only approximately 10% of those antibiotics administered to the hog or administered to the cattle is actually metabolized by the cattle or the hog. So that leaves approximately 80 to 90% that is being released from the animal into the excrement and into the environment. Once it hits the environment, we've already discussed the lagoon ponds, we've discussed the application of the waste, liquid waste and the sludge to the application field. So then you have a situation where you have terrestrial organisms that are being exposed to the antibiotics, but then you also have the liquid or dissolved antibiotics that are infiltrating through the soil. And then we're in a karst situation or a karst environment, such as near the Buffalo River and Mount Judy. We end up with infiltration down to the groundwater as well. So now we have antibodies that could possibly be transported in the groundwater, and then in situations such as karst, where you have a high level of exchange between groundwater and surface water, you also have the potential for those antibodies to reach the surface water. Biofilms. You guys may not have heard this term before, but biofilms are just a group of bacteria that are living together. They've attached to the surface, so they're no longer swimming around as you would normally expect, such as kind of what Chuck was talking about. They're looking at bacteria that are actually loose in the water, free swimming. I'm looking at attached bacteria here in this situation. So when you hear about biofilms, that's what we're talking about. So in their growth, they'll start off here in the water. Those single cells will drop out and attach to the surface. They'll congregate, they'll reproduce. So you have more bacteria that are actually growing at the surface, but then you also have different bacteria. So you're not looking at just one type of bacteria growing within a biofilm. You're actually looking at a whole system of bacteria with different functions as well. And so those bacteria are acting in symbiosis. So they're exchanging food with one another. So that's one advantage of being in the biofilm. But they also provide protection. Those biofilms, bacteria are also targeted by protozoa. They like to eat them. And so those protozoa are targeting these bacteria. They're harder to target when they're in biofilms together in colonies, and it's difficult to actually isolate any particular bacterial community. So those are two main things or main reasons, advantages that bacteria have forming biofilms. They are also able to self-preserve. So they exchange genetic information within these biofilms. That genetic information, such as, for instance, antibiotic resistance, can be exchanged between different communities of bacteria. Those different communities of bacteria then move on once that biofilm has matured, it's thickened to a point where it can't hold on anymore, and then you have the distribution of those bacteria, of the genetic information associated with that bacteria through the water system, through the flow system. So we conducted some laboratory studies. We set up small models within the laboratory using water that we collected from Big Creek, from the Buffalo River, and from springs. We've actually added some food to those small models, what we call them microcosms. We added food to them, so we added some nutrients, nitrogen and phosphorus. Then we also gave them a carbon source, something to kind of speed up or edge on the process of what they would normally be doing in the environment. We analyze them for carbon, nitrogen, phosphorus, and dissolved oxygen. So those are indicators of how active are the bacteria here in these systems. As I said, we add food. We use isotope labels kind of commonly. And what the isotope labels allow us to do is to trace the bacterial processes that are actually occurring. So things such as nitrification, denitrification, the bacteria removing nitrogen from the system, the bacteria adding different forms of nitrogen, removing carbon from the system. We use those types of tracers to say, well, this is how much of this is happening. We also conducted some antibiotic sampling this summer. This year, we're waiting still for the data to come back from the USGS Organic Geochemistry Lab in Orange, Kansas. But we're going to look at 4 different types of antibiotics, 4 different classes of antibiotics. So we're going to look at our macrolides, specifically erythromycin, and then we're going to look at tetracycline. Now, 2 major points about these. These are naturally produced antibiotic compounds. So there is already probably going to be a natural resistance within the environment that you will be able to detect. So that's one thing that we have to be careful about when we try to source and say,